Okay, I'm here again. Hello. After having identified the phytoplasmas that infect grapevine in Chile, was very important to understand how they spread in the vineyards. Therefore, epidemiological studies have been performed by our laboratory. Yes, you remember that the most widespread phytoplasma in Chile is the 3J, and this is a phytoplasma that we found not only in grapevine but in the several different hosts. And we in some vineyards we found it in the grapevine plants, but also in convolvulus several weeds, also called convolvulus arvensis, Polygonum aviculare, Brassicarapa, and Gallego officinalis. We performed transmission trials with two leaf hoppers that we found positive to the same 3J phytoplasmas, and we found that Paratanus exitiosus and Bergallia valdiviana are vectors of these phytoplasmas. And then in the here you can see the symptoms in periwinkle and grapevine plants that we use in transmission trials. You can see the yellows, which is broom in periwinkle plant, virescence, phyllody, double nodes, short internodes, and generally malformation of the leaves. Well, this was the first step of our investigation, our research, but the research has not stopped, as it seems very likely that other species of insect and reservoir plants were involved in the 3J phytoplasma spread. We captured insect and collected plants in 3 different vineyards in Chile. The first one cultivated with Pinot Noir in Casablanca, Valparaiso regions. The other one with Cabernet Sauvignon and Marchigüe in O'Higgins regions, at the south of Santiago, and at the south of O'Higgins regions, the Maule region, San Javier, with the Carmener variety. In this, the insect were collected or were captured inside the vineyards, but also in the areas surrounding the vineyard itself. And each vineyard was visited monthly, and the insect capture was carried out with a sweeping entomological net oriented towards the weeds. Captured individuals were placed in test tubes with a cotton plug. Then in the lab, the insects were identified by the optical microscope, and during the same fields, we collected, as I told you before, some weeds as malva with leaves deformation and short growing or reducing growing or rubus ulmifolius. Then insects and plants have been analyzed for the presence of phytoplasma following the nested PCR protocol with the primer P1, P7 and nested with R16F2N, R2. Then we cloning and sequencing the amplification product. We performed bioinformatic analysis and RFLP. We used other gene, tough gene, to confirm the identification of the phytoplasmas. These are the results. As you can see, several leaf hopper that we found in the vineyard and the vicinity resulted positive to 3J phytoplasma generally, but 2, specifically Amplicephalus curtulus exitianus obscurinervi, was positive to new phytoplasma, the 15 and 11 phytoplasma. This is a great news for us in this research. The insect found positive Amplicephalocurtulus. This is the imagine, the picture of this insect, which was positive for 3J11 15 phytoplasma. Amplicephalocurtulus positive to 3J. Exitianum oscurinervis positive to 11, the subgroup 11 phytoplasma, and Bergallia SP positive to 3J. Then in the weights, we found again 3J in several weights associated to the raven or near to the vineyards, and Dactylis glomerata cynodondattyle lolium was found positive to the phytoplasma 11 in the Marchigue vineyard. Very interesting, this vineyard in Marchigue. This is our malva and brassica rapa found positive to 3J. No symptoms, only in malva some deformation of the leaves. And other plants, no weeds, no grapevine, were found positive to 3J phytoplasma, always in Marchigue vineyard, and specifically Rosa and Rubus ulmifolius. These two plants that normally are used as near to the vineyard normally. And these are the symptoms observed normally, which is broom or reducing or growing, the Rubus ulmifolium and Rosa. In conclusion, plants, we observe the plants other than grapevine play a relevant role as a reservoir of the phytoplasma 3J. We found a lot of plants other than grapevine positive to this phytoplasma. This is the first report of the 16SR11 phytoplasma detected in insect, Exitiano scurinervi, and the weeds Dactylis glomerata, cynodondattyle lolium sp. And this is the first report of the phytoplasma 16SR15 detecting in insect Amplicephalocurtulus, and the phytoplasma 16 SR11 and 16 SR15 have not been detecting any plant species of economic interest in Chile at the moment. Thus, an intense sampling of vegetable material is taking place in the locality in which the phytoplasma were found, the vineyard present in Marchigue, but not only in the vineyard, because we are searching the phytoplasma in other species too. The identification of the subgroup 11, the group 11 and 15 ribosomal group are in progress. We don't know at the moment which are the subgroup associated with this ribosomal group. We are working to found the ribosomal subgroup, as well as the transmission trials with the new insect species that we captured. As a general conclusion, the insect is very important that the insect captured in the Chilean vineyard do not represent a direct problem for the crop, generally, for the grapevine. The insect generally live associated with the spontaneous plant species and only occasionally feed on the grapevine plants, transmitted phytoplasmas. Apparently, the presence of few individuals contributed effectively to the spread of phytoplasma in the vineyards, even more when infected spontaneous plants are found in the field. Therefore, the indirect damage of that the insect caused to the grapevine should not be underestimated. They are very important because transmit the 3J phytoplasma. Spontaneous infected plants present in or in the vicinity of the vineyard are important reservoir of the 3J phytoplasma. Thank you very much for your attention.